How can a simple organism buried deep in the pitch-black rainforest floor know exactly when the night insects will take flight? Deep within the Brazilian rainforest, a master of evolutionary deception lies in wait. The coconut forest is a transitional biome spanning 4 states in northeastern Brazil, nestled between the humid density of the Amazon and the dry, thorny expanse of the Cerrado savanna. It is one of the lesser-known biomes on the planet. Tall babaçu palms dominate the landscape, their fallen fronds and husks accumulating on the forest floor in slow, perpetual decay. But beneath that canopy, where the sunlight barely reaches and the air sits heavy and wet, something extraordinary happens after dark. Most people wandering into this forest at night would be struck first by the darkness, the kind of darkness that your eyes never fully adjust to, where the absence of light feels like a presence in itself. The canopy seals out the stars, the undergrowth swallows what little ambient glow might filter down. You are, for all practical purposes, blind. And yet, on certain fallen coconut palm husks, on certain decaying logs and leaf litter patches scattered across the forest floor, something glows. Not in the uncertain way that you might squint at a distant firefly and wonder if you imagined it. This glow is steady, persistent, and once your eyes have adjusted to it genuinely, breathtakingly beautiful. A cool, vivid yellow-green light pulses from the darkness, seemingly from nowhere, hovering just above the forest floor like something that has escaped from a science fiction novel or a fever dream. It is a light that does not flicker in the wind and is completely alive. The organism producing this extraordinary light is Neonothopanus gardneri, a bioluminescent mushroom first formally described to Western science in 1840 by the English naturalist George Gardner, hence the species name, who encountered glowing coconut husks near the town of Natividad in the Brazilian state of Tocantins. Gardner's initial account, published in the botanical literature of the time, was greeted with the mixture of fascination and skepticism that tends to greet any discovery that sounds, frankly, too good to be true. A glowing mushroom in the jungle? It sounds like folklore. It sounds like the kind of thing that indigenous communities describe in their traditional stories and that scientists politely file under probably exaggerated. Except it wasn't. Not even slightly. Neonothopanus gardneri belongs to the family Marasmiaceae, part of the Omphalotus lineage of bioluminescent fungi, though none of its relatives are quite as dramatically visible, none quite as persistently luminous, and none quite as behaviorally complex as this particular Brazilian resident. It grows at the base of several species of dwarf palm, including the pindoba palm, the piaçava palm, and the babaçu palm, feeding on their decaying fronds and husks. These palms dominate vast swaths of the transitional forest between the Amazon basin and the Brazilian Cerrado savanna. The relationship between fungus and palm is not incidental, it is foundational. N. gardneri has specialized over millions of evolutionary tinkering to exploit precisely this substrate, to thrive in precisely this niche, and, as researchers would discover only in the 21st century, to exploit the creatures that share this niche in ways that are almost impossible to believe. But before we get to how it's manipulated, we need to understand light itself, because the whole game lies in the light. Let's take a step back, because if you spent any time poking around forests at night, and if you're a mushroom enthusiast in the United States, there's a decent chance you have. You might already know that bioluminescent fungi are not exactly rare. There are 132 confirmed bioluminescent fungal species known to science, as of the most recent comprehensive count, a number that has more than doubled over the past 15 years as researchers keep finding more, and mycologists suspect the true number is considerably higher, given how many fungal species remain formally undescribed. They span multiple families and multiple continents. Some are dramatic and visible. Many are subtle enough that you'd have to sit in genuine darkness for 20 minutes, eyes fully adapted, nose almost touching the substrate, to see anything at all. In North America, you've probably got a few in your own backyard, metaphorically speaking. Panellus stypticus, the bitter oyster, glows with a faint greenish light from its mycelium and sometimes its fruiting bodies. If you've found it on decaying oak logs in the eastern United States, and it's widespread enough that many of you likely have, and if you've ever taken it into a truly dark room and waited long enough, you might have seen the faintest suggestion of luminescence. Omphalotus olearius, the jack-o'-lantern mushroom, famously glows from its gills, a fact that has contributed to more than a few emergency room visits when people mistake it for the golden chanterelle and have a very unpleasant evening. The glow is real, though again you need genuine darkness to appreciate it. Mycena chlorophyos puts on a genuinely impressive display in parts of Asia and Australia. And scattered across the world's tropical forests, various species of Mycena, Panellus, Philoboletus, and others add their quiet luminescence to the night.
So, bioluminescence in fungi is not the exceptional novelty it might first appear. But what is it, exactly? How does a mushroom, an organism without a nervous system, without eyes, without any apparent capacity to experience the light it produces, manage to generate it? The chemistry is surprisingly elegant. Fungal bioluminescence, like that of fireflies and deep-sea fish, relies on a reaction between a light-emitting molecule called a luciferin and an enzyme called a luciferase. But here's where it gets interesting. The luciferin used by fungi is chemically distinct from those used by other bioluminescent organisms. It is not the same system that a firefly uses. Fungi evolved their light-producing chemistry independently, which, when you think about it, tells you something important. Evolution arrived at bioluminescence multiple times in multiple kingdoms of life through different biochemical routes. That kind of convergent evolution is usually a sign that whatever the trait does, it is genuinely useful enough to be worth the metabolic investment of developing it. In fungi, the luciferin is a compound derived from caffeic acid, a precursor that is itself a product of the same biochemical pathway that breaks down the lignin in decaying wood, which makes perfect sense given that most bioluminescent fungi are wood decomposers. The reaction cycle works something like this. Caffeic acid is converted into the active luciferin, which then reacts with oxygen in the presence of the luciferase enzyme, releasing energy as light in the process. The spent luciferin is then recycled back into its precursor form, and the cycle begins again. It is, in essence, a self-sustaining biochemical lantern, powered by the same metabolic processes the fungus uses to break down its food. Now, the critical question, why? What does a fungus gain from glowing? This question turns out to be considerably more contentious than you might expect, and it has occupied mycologists, ecologists, and evolutionary biologists for a remarkably long time. The honest answer is that we are still not entirely certain, and that different bioluminescent species may glow for different reasons. In some cases, the luminescence may simply be a metabolic byproduct, a consequence of the chemistry of wood decay that costs the organism relatively little and confers no particular advantage, which is why it persists without being strongly selected against. This explanation is probably adequate for species with very faint, inconsistent luminescence. But for Neonothopanus gardneri,
which produces one of the brightest and most consistent bioluminescent displays in the entire fungal kingdom. The byproduct explanation starts to feel deeply unsatisfying. Something bright enough to read by, and researchers have confirmed that gardneri is, in ideal conditions, genuinely bright enough for that. Something that glows steadily all night long, every night, that has evolved a biochemical clock to regulate its luminescence in synchrony with the cycles of night insects. That is not a byproduct. That is a strategy. Here is where Neonothopanus gardneri begins to reveal its true sophistication, and where the story shifts from charming natural history into something that should make the hair on the back of your neck stand up.
In 2015, a team of researchers led by Cassius Stavani at the University of São Paulo published a study in the journal Current Biology that changed the way scientists think about bioluminescent fungi. What Stavani's team discovered was this: Neonothopanus gardneri does not glow all the time. It glows at night. Now, you might think, well, obviously, it's a night thing. We knew that. But you would be underestimating how genuinely remarkable this observation is, because the mechanism behind it is not what you might assume. The naive explanation would be that the fungus simply responds to darkness, that when light levels drop below a certain threshold, some photosensitive molecule triggers the bioluminescence reaction, and when light levels rise again in the morning, it switches off. Simple stimulus and response, nothing to write home about. But that is not what happens. What Stavani and his colleagues discovered is that gardneri maintains its luminescent rhythm even when kept in constant darkness. Even when the forest disappears and the fungus is placed in a laboratory with no light cues whatsoever, it continues to glow at night and dim during the day, following a cycle that it is tracking internally without any external reference point. Neonothopanus gardneri has a circadian clock. Let that sink in for a moment. This fungus, this organism that we have historically lumped with plants, that lacks a brain, a nervous system, eyes, or any sensory apparatus recognizable to us, maintains a biological clock accurate enough to predict the transition between day and night based on internal biochemical cycles alone. Temperature fluctuations help calibrate it, keeping it synchronized with the real world, even without light cues. But the clock itself runs independently. This is not unique to fungi. Circadian rhythms have been documented across virtually all domains of life, from bacteria to humans. But finding them in a mushroom that uses them to time its bioluminescence to the flight patterns of specific insects is something else entirely. Because the clock is not just telling the fungus what time it is, the clock is coordinating a manipulation. The researchers found that the peak luminescence of this mushroom corresponds almost exactly with the peak activity of the insects that visit it. The fungus is not simply glowing at night in some general sense. It is glowing most brightly at precisely the times when the insects most likely to visit it are most active. It has tuned its internal clock to match the behavioral rhythms of the creatures it needs. And what does it need them for? To understand what Neonothopanus gardneri is doing with its light, we need to think carefully about the problem it faces and the solution it has evolved. The fungus, like all fungi, reproduces via spores. Spores are microscopic, lightweight packages of genetic material, each one a potential new individual, each one needing to travel from the parent organism to a new substrate where it can germinate and grow. Many fungi disperse their spores passively. They simply release them into the air and let wind currents carry them wherever chance dictates, a strategy that works reasonably well if you produce enough spores and if air movement is sufficient. But this mushroom lives on the floor of a dense rainforest. Air movement at ground level in a mature tropical forest is minimal. The canopy absorbs most of the wind energy before it ever reaches the understory. Passive airborne dispersal is not a reliable strategy in this environment. The fungus needs an alternative. Plants offer nectar, fruit, or pollen rewards to attract insects, birds, and mammals, which then carry pollen or seeds to new locations as they go about their lives. The plant gets dispersal, the animal gets food. Everybody wins. Fungi have explored variations of this theme throughout their evolutionary history. Many species produce attractive scents or food rewards that draw insects or other invertebrates, which then pick up spores on their bodies and carry them to new locations. Some fungi have gone further, evolving elaborate and often genuinely bizarre mechanisms to attract specific dispersal agents. Neonothopanus gardneri attracts insects with light. The bioluminescence acts as a lure, drawing in arthropods, beetles, flies, ants, cockroaches, and various other invertebrates that are attracted to the glow. Exactly why insects are attracted to the fungal light is a question that ecological researchers are still working to fully answer. But the best current hypothesis is that the light mimics other light sources in the insect's environment that are genuinely rewarding, a phenomenon known as sensory exploitation or sensory trap, in which a signal takes advantage of a pre-existing sensory bias in the receiver. Night-flying insects in tropical forests navigate by, orient toward, and are naturally attracted to various natural light sources: the moon, gaps in the canopy, the bioluminescence of other organisms. Some of them are attracted to the light because they use it for navigation, and the steady glow of the fungus confuses their orientation systems, causing them to spiral inward toward the source, the way moths circle a flame. Others may be attracted because light sources in the forest environment often indicate food, fruit, flowers, or other organisms gathered around a light-emitting creature, and evolution has wired them to investigate. Whatever the specific mechanism, the result is the same. Insects come to the glowing mushroom. And when they arrive, they walk across the surface of the fruiting body, through the gills or pores where the spores are concentrated, and they pick those spores up on their legs, their bodies, their mouthparts. Then they leave carrying N. gardneri's genetic material with them into the forest, to be deposited, perhaps, on a fresh palm husk somewhere that the fungus could never reach on its own. This is, in its broad outline, a story about mutualism or commensalism. The fungus benefits, the insect may or may not, depending on what, if anything, it receives in return. But the word manipulation in our title implies something more active, more coercive than simple attraction. So let's talk about what happens to the insects that visit and about what the fungi might actually be doing to their behavior. The idea that fungi can manipulate insect behavior is not, at this point, a controversial one. If you are at all plugged in to the popular science world, you have almost certainly heard of Ophiocordyceps, the so-called zombie ant fungus, a parasitic fungus that infects carpenter ants, hijacks their nervous systems, forces them to climb to a precise height above the forest floor, and bite down on a leaf vein at a specific time of day, then kills them and erupts from their bodies to release spores from the optimal dispersal height. It is one of the most dramatic and well-documented examples of behavioral manipulation in the natural world, and it has captured the popular imagination with good reason. The Ophiocordyceps example is extreme. The fungus is a lethal parasite that destroys its host to complete its life cycle. What Neonothopanus gardneri appears to do is more subtle and in some ways more interesting, because it does not necessarily require killing its visitors. The well-documented part of the story is this:
A 2015 study led by researchers at the University of São Paulo built acrylic resin replicas of N. gardneri fitted with green LEDs matching the wavelength and intensity of the real bioluminescence and placed them in the forest alongside identical but unlit decoys, all coated in sticky glue. Over 5 nights, the illuminated decoys caught significantly more insects, flies, beetles, wasps, and true bugs than the dark controls. That is solid, peer-reviewed evidence that the light itself functions as a lure.
What the experiment does not show, and what I want to be careful not to imply, is that scientists have tracked individual insects walking across the mushroom and documented changes in their locomotion once they land. That specific kind of behavioral study, the close-up tracking of an insect's movement pattern on the fruiting body itself, is not something I could find solid published evidence for. What we know for certain is the recruitment side of the story. The light draws insects in from a distance. What happens in the seconds after they land, whether they linger, whether something about the mushroom's surface or chemistry keeps them there longer than they otherwise would stay, is a more open question and a fair place for honest speculation rather than a settled fact. What we do know is that the outcome is strikingly effective. N. gardneri is widely distributed across its habitat range, successful enough to be encountered regularly by observers who know where to look, and the spore dispersal mechanism it has evolved appears to be working. The fungus is winning at the game of reproduction. And it is doing so with light, chemistry, and timing, a toolkit that took millions of years to assemble and that we are only beginning to understand. Now, here is a question that I want to pose to you because I think it is one of the most fascinating questions in all of mycology and one that does not get nearly enough attention. How old is the light? When did Neonothopanus gardneri first begin to glow? When did bioluminescence first appear in fungi at all? And what can the evolutionary history of fungal luminescence tell us about the world those early glowing organisms inhabited? The fossil record for bioluminescence is, as you might imagine, essentially non-existent. Light does not fossilize. The biochemical machinery that produces it leaves no reliable trace in ancient rocks. We cannot point to a chunk of Cretaceous amber and say, here is a glowing mushroom frozen in time, exactly as it appeared when the dinosaurs walked past it in the darkness. The history of bioluminescence must be reconstructed not from fossils, but from genetics, from the comparative analysis of the genes responsible for the luminescent reaction in species alive today. In 2018, a landmark study by a team including researchers from Russia, Brazil, and the United States, published in the Proceedings of the National Academy of Sciences, traced the evolution of bioluminescence across all known luminescent fungal lineages using a technique called ancestral sequence reconstruction. What they found was simultaneously clarifying and humbling. Bioluminescence in fungi appears to have arisen once, a single evolutionary event, sometime in the distant past, in the common ancestor of all bioluminescent fungal species alive today. That single origin event gave rise to the entire diversity of glowing fungi we see now, from the faint luminescence of Panellus stypticus on your local oak log to the blazing display of N. gardneri in the Brazilian night. All of that diversity traces back to one ancestor that, for reasons we can only speculate about, developed the chemistry of light. The timing of that origin event is estimated to be approximately 160 million years ago, the Jurassic period, the age of dinosaurs. The continents were arranged differently. The climate was warmer and wetter. The forests were dominated by conifers and cycads rather than flowering plants, and the mammals that would eventually give rise to us were small, nocturnal, and probably terrified of most things.
And somewhere in the dark forests of that ancient world, a fungus began to glow. What triggered it? What advantage did that first glowing ancestor gain? Here is where the story becomes genuinely speculative, and where I want to invite you to sit with the uncertainty for a moment, rather than rushing past it, because the uncertainty itself is instructive. The hypothesis that best fits the current evidence is the one we have already explored, that bioluminescence evolved as an insect attractant for spore dispersal, and that the insects of the Jurassic period exerted selective pressure on fungi that could attract them more effectively. The biochemistry of the luminescent reaction, which runs off the same metabolic pathway as lignin degradation, may have made the initial appearance of faint luminescence relatively easy to achieve. A small mutation in an enzyme, a slight modification to an existing pathway, and suddenly a mushroom that glowed ever so dimly. If even that faint glow attracted insects and improved spore dispersal, natural selection would have rewarded it, and over generations the glow would have intensified and refined. Over 160 million years of evolution, that original faint chemical signal has been shaped, tuned, and in some lineages completely revolutionized into the extraordinary biological light show that gardneri puts on every night in the Brazilian forest. The circadian clock that synchronizes its glow with insect activity has been calibrated over enormous stretches of evolutionary time by the selective pressure of insects that were more active at some hours than others. The chemistry has been optimized, the timing has been perfected. It is, in the most literal sense, a 160 million year arms race between a fungus and the insects of its world, played out in darkness, measured in light. Let's get practical for a moment, because I know some of you are already Googling bioluminescent mushrooms near me and trying to figure out if there is any realistic chance of experiencing something like what I have been describing. The answer depends very much on where you are, but it is better than you might think. Neonothopanus gardneri itself is restricted to Brazil, specifically to the Mata dos Cocais and the transitional zones into the Cerrado. And if you want to see it in person, you are looking at a trip to Piauí, Maranhão, Goiás, or a handful of other Brazilian states where the babaçu palm grows abundantly and the forest fragments are large enough to maintain viable populations. That is not a trivial undertaking, and I will not pretend otherwise, but it is possible, and mycologists and nature photographers who have made the journey describe it as one of the most genuinely transformative experiences available to a naturalist. If the Brazilian rainforest is on your bucket list for any reason, adding this mushroom to your itinerary would not be a bad idea. For those of you in the continental United States, however, there are real and genuinely accessible bioluminescent fungi worth seeking out. Let me give you a practical guide. Pinellus stypticus, the bitter oyster, is your most reliable option across much of the eastern United States. It grows on decaying hardwood logs, particularly oak, beech, and birch, and it is common enough that if you spend any time in eastern deciduous forests, you have almost certainly walked past it without knowing what you were missing. The glow is in the mycelium, which means you are more likely to see it in a fresh, actively growing specimen than in an old, dried-out one. To see the luminescence, you need genuine darkness, not just a dim room, but a properly dark space, with your eyes fully dark adapted for at least 15 to 20 minutes. On the forest floor, on a moonless night, away from any light pollution, a healthy patch of P. stypticus mycelium will reward your patience with a faint but real blue-green glow. It is subtle, it is beautiful, and knowing that you are seeing the same biochemical reaction that 160 million years ago was just beginning its evolutionary journey toward the blazing displays of N. gardneri, that context makes it genuinely moving.
Omphalotus illudens and Omphalotus olearius, the jack-o'-lantern mushrooms, are the most dramatic bioluminescent display readily available in North America. O. illudens is the eastern species. O. olearius occurs in the west and is also widespread in Europe. These are large orange chanterelle-like mushrooms that grow in clusters at the base of trees or from buried roots. They are saprotrophic on wood, so you will find them associated with stumps, dying trees, or areas where old trees have been removed. The gills glow. On a good specimen, in genuine darkness, the glow is visible almost immediately after dark adaptation, and it is genuinely impressive. Not quite the blazing beacon of N. gardneri, but real and unmistakable. Fair warning, these mushrooms are toxic. Do not eat them. And if you collect chanterelles, please learn to distinguish them from jack-o'-lanterns. The growth habit is a good starting point, since chanterelles are solitary and mycorrhizal, growing directly from soil, while jack-o'-lanterns cluster from wood. Mycena species are worth investigating, particularly in the Pacific Northwest, where the damp, woody habitat suits several potentially bioluminescent species. The taxonomy of glowing Mycena is still being worked out in North America, and there are confirmed and suspected luminescent species in the genus on this continent, though I want to be precise here. Mycena luxaterna itself is not one of them. That species is endemic to a single very limited stretch of Atlantic forest in São Paulo state, Brazil, and has never been documented in North America. If you find delicate, small-capped mushrooms growing on decaying wood in the Pacific Northwest, take them into the dark anyway. You may be surprised by what you find, even if it's not that particular species.
The practical tips for seeing fungal bioluminescence are worth spelling out clearly. First, find your specimen during the day so you know where it is. Trying to navigate a dark forest looking for a faint glow from scratch is an exercise in frustration. Second, wait. Your eyes need genuine dark adaptation. 15 minutes minimum. 20 is better. During this time, resist every temptation to check your phone. One glance at a bright screen will reset your adaptation entirely. Third, get close. Fungal bioluminescence is not bright by any standard. You need to be within arm's reach, ideally with your face near the surface. Fourth, choose the right conditions. Bioluminescence is temperature sensitive. Most fungal luminescent reactions slow down in cold conditions and are more active in warmer temperatures. A mild, humid night is better than a cold, dry one. Fresh specimens glow more brightly than old ones. Fifth, and most importantly, manage your expectations, and then let reality exceed them anyway. Because even faint fungal bioluminescence, seen in its proper context in a living forest at night, knowing what you know about the 160 million year story behind the light, is not a disappointment. It is a privilege. Neonothopanus gardneri is extraordinary, but it exists within an extraordinarily rich context of bioluminescent fungi across the world, and I want to take some time to explore that broader world, because each species adds something to our understanding of what the light is doing and why it evolved. Mycena chlorophyllus is perhaps the most photographed bioluminescent mushroom in the world, and with good reason. It grows in large numbers on decaying wood in tropical Asia and Australia, and it puts on a display vivid enough that photographs taken in total darkness with nothing but the mushroom's own light as the illumination source look genuinely surreal. The glowing caps cluster in groups of dozens or hundreds, creating scenes that look like something from the imagination of a fantasy illustrator. It is common enough in parts of Japan that bioluminescent mushroom walks have become a tourist attraction, with guided nighttime forest hikes organized around peak fruiting season. Pinellus stypticus, which we have already mentioned as your best bet in North America, has a fascinating geographic quirk. The North American populations of this species are significantly more bioluminescent than European populations. The reason for this difference is not fully understood, but it has been used to explore the genetics of bioluminescence, since the two populations are closely enough related to allow direct genetic comparisons. What genes are present in North American P. stypticus that are absent or non-functional in European populations? Understanding those differences helps researchers map the specific genetic machinery of fungal luminescence, work that has implications far beyond mycology. Phylobolets manipularis is a Southeast Asian species that is remarkable for the consistency and brightness of its glow relative to its small size. The entire fruiting body, cap, stalk, and gills luminesces, creating a ghostly but vivid total organism display.
And then there is the remarkable case of Mycena lux eterna, which was formally described in 2010 from specimens collected in the Atlantic forest of Sao Paulo state, essentially in N. gardneri's backyard, though in a completely different biome, by the same research group that has been central to modern bioluminescence studies. Its specific epithet, lux eterna, means eternal light, a reference to the Latin liturgical phrase lux aeterna luceat eis, may eternal light shine upon them, which the describing authors noted as the inspiration, since the mushroom's tiny glowing caps in the dark forest called the phrase to mind. That a group of scientists chose a name with that kind of resonance is, I think, a beautiful illustration of how far beyond mere data collection the study of bioluminescent fungi takes you. Across all of these species, the biochemical pathway is essentially the same. The caffeic acid-derived luciferin, the luciferase enzyme, the self-regenerating cycle, but the expression is wildly different. Some glow only from mycelium, some from fruiting bodies, some from both. Some glow blue-green, some glow more purely green. Some glow steadily, some have diurnal rhythms. The same basic chemistry has been shaped by evolution into a remarkable diversity of light shows, each presumably tuned to the specific ecology of the organism that produces it. One fascinating area of ongoing research concerns the relationship between bioluminescence and the broader chemical ecology of fungi. If the light is primarily an insect attractant for spore dispersal, you would expect to find some correlation between bioluminescence and ecological context. Specifically, you might expect bioluminescent species to be more common in habitats where passive spore dispersal by wind is less effective. Dense tropical forests with their still understory air should theoretically favor insect-mediated dispersal and therefore bioluminescence. And indeed, when you map the distribution of confirmed bioluminescent species worldwide, there is a noticeable bias toward tropical and subtropical forest environments, and toward species that grow on the forest floor or at low heights on decomposing woody material, exactly the habitat profile where air-mediated dispersal would be least effective. This is not conclusive evidence. Correlation is not causation, and there are confounding factors, but it is consistent with the insect attractant hypothesis. There is also a small but intriguing cluster of bioluminescent species in temperate and even boreal forests, which complicates the simple picture somewhat and suggests that additional selection pressures or additional functions for the light may be at play in certain ecological contexts.
Some researchers have proposed that in temperate species, bioluminescence might function not primarily as an insect attractant, but as a warning signal, aposematism in chemical terms, advertising toxicity or unpalatability to potential grazers. This would explain why some species glow from mycelium only and not from fruiting bodies, since mycelium is more vulnerable to grazing damage than the tougher tissues of mature fruiting bodies.
The truth is probably that bioluminescence in fungi serves multiple functions simultaneously in different species and ecological contexts, and that the evolutionary history of fungal light is considerably more complex and multifaceted than any single neat hypothesis can capture. Which is exactly the kind of answer that makes a scientist uncomfortable and a natural historian happy. There is something philosophically vertiginous about Neonothopanus gardneri once you sit with it long enough. Consider what this organism knows, or rather, what information it embodies, encoded not in neurons or language or memory in any sense we recognize, but in the biochemistry of its cells, in the architecture of its circadian clock, in the carefully calibrated wavelength of light it produces. It knows when night will fall. It knows when the insects will fly. It knows how to attract them, how to load them with spores, how to ensure they carry those spores somewhere useful. It has encoded the rhythms of an entire ecosystem into its molecular machinery, the temperature cycles of a Brazilian day, the activity patterns of insects whose ancestors coexisted with its own ancestors for millions of years, the physics of light propagation through a dense forest understory. It does not know these things the way you know that the sun rises in the east or that rain falls down. It knows them the way your heart knows to beat, the way your immune system knows a pathogen, not through conscious awareness, but through the accumulated wisdom of evolutionary time, compressed into biological structure and expressed without deliberation. We tend to reserve the word knowledge for minds. We tend to think of intelligence as requiring something recognizable as thought. But there is a genuine argument made seriously by philosophers of biology, and not just by enthusiastic science communicators, that the kind of environmental information encoded in an organism's adaptation to its niche represents a form of knowledge that predates and in many ways surpasses what we tend to celebrate in our own cognition. Gardneri has been solving the problem of spore dispersal in a dark forest for 160 million years. Human civilization has been solving the problem of surviving in complex societies for maybe 50,000. In terms of successful implementation of a working solution to a specific problem, the fungus has us beaten by several orders of magnitude. I am not suggesting that mushrooms are smarter than people. I am suggesting that the way we think about intelligence, knowledge, and even consciousness may be too narrow to fully appreciate what is happening in the living world around us, and that spending time with organisms like gardneri has a way of usefully unsettling those assumptions. The mycelial networks we have come to understand better over the past 2 decades, networks that process information, solve problems, communicate across distances, and coordinate the behavior of the forest around them, are not intelligent in the way that we are intelligent, but they are something. They respond, integrate, remember in their way, and make functional decisions that have consequences. Calling that nothing because it doesn't look like what we recognize in ourselves seems like a failure of imagination rather than a rigorous scientific position. Neonothopanus gardneri, with its circadian clock ticking in the darkness, its biochemical lantern drawing insects toward it across millions of evolutionary years of refinement, offers a window into a kind of knowing that is genuinely alien to us and genuinely worth knowing about. Let's bring this home. Let's talk about why any of this matters beyond the sheer gorgeous strangeness of a glowing mushroom in a dark Brazilian forest. The biochemistry of fungal bioluminescence
has attracted serious attention from biotechnologists, and for good reason. The self-regenerating luciferin luciferase system of gardneri and its relatives, a system that produces light continuously without external chemical supplementation, driven by the organism's own metabolism, is considerably more practical as a research tool than many existing bioluminescent systems used in laboratory biology. Currently, the most widely used bioluminescent reporter system in molecular biology is derived from fireflies. The loop gene, encoding firefly luciferase, which is inserted into cells as a reporter to track gene expression. It works beautifully, but it requires the external addition of the firefly luciferin, D-luciferin, which is expensive and which the cells cannot make themselves. This limits the applications of the system in living organisms, since you cannot easily get D-luciferin into every cell of a living mouse or a growing plant in a controlled way. Fungal bioluminescence does not have this limitation. Because the fungal system is self-contained and the luciferin is derived from a metabolite that most cells already produce or can be engineered to produce, it can theoretically be expressed in a wide variety of organisms, from bacteria to plants to mammalian cells, and generate light continuously from within without external supplementation.
Researchers have already demonstrated this in practice. In 2020, a team from Russia and Brazil reported success in engineering tobacco plants to express the complete fungal bioluminescence pathway, resulting in plants that glow continuously throughout their lives with visible light, the first true glowing plants that generate sustained naked-eye visible bioluminescence using only their own metabolism. The results were published in Nature Biotechnology and generated exactly the amount of public excitement you would expect from photographs of softly glowing tobacco leaves. The practical applications range from the immediately useful bioluminescent reporter systems for tracking gene expression in living organisms without external luciferin, or for monitoring plant health and stress responses, to the genuinely futuristic, including the possibility of bioluminescent crops that signal when they need water, fertilizer, or treatment for disease by changing their glow pattern. Imagine a field of wheat that lights up at night to tell you which plants are under fungal attack. The idea seems like science fiction, but the biochemistry is already in hand. The engineering is being refined. Beyond biotechnology, the study of gardneri and other bioluminescent fungi is contributing to our understanding of circadian biology, chemical ecology, and evolutionary genetics in ways that have implications across multiple fields. The circadian clock of this mushroom, tuned to the rhythms of tropical insects over millions of years, shares molecular features with the biological clocks of animals, including humans, and studying it illuminates the deep evolutionary roots of timekeeping in living systems.
The chemical communication between the fungus and its insect visitors, whatever form that communication ultimately turns out to take, is part of the vast and still poorly understood landscape of cross-kingdom chemical ecology, in which the volatile compounds and secondary metabolites produced by fungi shape the behavior, physiology, and evolution of the organisms that live alongside them. Understanding those interactions matters for agriculture, for medicine, for conservation biology, and for our fundamental understanding of how ecosystems work. And the bioluminescence itself, once a curious footnote in mycological literature, is now recognized as a window into the evolutionary history of an entire kingdom of life, a history that stretches back 160 million years, and that is still actively unfolding in the dark forests of Brazil every single night.
I want to speak directly to the mushroom enthusiasts in the audience for a moment, because you are the people who have the most direct access to this world, and I think that access is worth celebrating and encouraging. The study of bioluminescent fungi is, by the standards of professional mycology, a young and under-resourced field. There are more species yet to be formally described than have been formally described. There are questions about the ecology, chemistry, and evolutionary history of these organisms that could be answered by careful, systematic observation in the field. Observation that professional researchers, constrained by funding, time, and geography, cannot always provide.
Citizen science contributions to mycology have been transformative over the past 2 decades. Platforms like iNaturalist have made it possible for observations made by amateur naturalists to contribute meaningfully to our understanding of fungal distribution, phenology, and ecology. If you observe a bioluminescent fungus, any bioluminescent fungus, photograph it, using a tripod and a long exposure in darkness, not a flash. Record the substrate, location, date, and conditions, and upload it to iNaturalist with as much detail as possible. Those observations matter. They have been used in published research. They are being used right now. If you want to go deeper, the work of Cassius Stevani's group at the University of São Paulo is the center of gravity in bioluminescent fungal research, and their publications, many of which are freely available online, are some of the most readable and well-illustrated scientific papers you will find on any topic. The 2015 Current Biology paper on N. Gardneri's circadian clock is a good starting point. The 2018 PNAS paper on the evolution of fungal bioluminescence is essential reading for anyone who wants to understand where the light came from. For North American bioluminescence, David Hibbett's work on the phylogenetics of the Omphalotaceae, the jack-o'-lantern family, provides crucial evolutionary context. And if you have never read Merlin Sheldrake's Entangled Life, stop whatever you are doing and read it, because it is the finest accessible treatment of the fungal world available to a general audience and will change the way you see every forest you walk through for the rest of your life. For those of you interested in biotechnology applications, the 2020 Nature Biotechnology paper by Mitiuchkina and colleagues on bioluminescent plants is freely accessible and extraordinarily well written for a scientific paper. It includes photographs that you will want to look at for a long time. And for the adventurous among you who want to see N. gardneri in person, it is possible. The Brazilian states of Piauí and Maranhão are the most accessible locations where the species is known to fruit reliably. The fruiting season corresponds roughly with the wet season, from October to April, with peak displays often in December and January. Conservation organizations working in the Mata dos Cocais region can often provide guidance on locations and timing. This is not a casual day trip, but it is a pilgrimage worth considering. There is a dark stretch of rainforest somewhere in eastern Brazil at this very moment where a coconut palm husk is slowly, silently, imperceptibly decaying. Deep within it, a network of hyphal filaments, each 5 times thinner than a human hair, is threading its way through the fibrous material, breaking it down, extracting from it the nutrients and energy that will fuel a fungus through another season. The fungus is Neonothopanus gardneri. Its internal clock is running. It knows, in the only way it knows anything, that nightfall is approaching. The machinery of bioluminescence is primed and ready. The enzymes are in place. The luciferin is cycling through its molecular dance, waiting for the command to release its energy as light.
As darkness falls in the forest, as the last birds go quiet, and the first insects of the night begin to stir in the canopy, the fruiting bodies of gardneri emerge from the forest floor and begin to glow. Somewhere above them, a beetle lifts off from a branch and begins its nightly circuit of the forest understory, navigating by the faint light gradients of the clouded sky. And then it sees something brighter, something steadier, something that in 160 million years of evolution, it has been conditioned by countless generations of natural selection to approach. It descends, it lands, it walks across the surface of the glowing mushroom, through the gill spaces where the spores wait, and without knowing it, without anything that we would recognize as intention or decision, it becomes part of the oldest ongoing collaboration between a fungus and an insect on the planet. It carries the future of gardneri in the microscopic passengers clinging to its legs and body as it lifts off again into the Brazilian night. The mushroom glows on. It does not know that researchers on the other side of the planet have used its biochemistry to make plants glow. It does not know that the papers written about it have circulated through dozens of countries and that thousands of people have spent time staring at photographs of its light, their faces illuminated by the glow of their phone screens in darkened rooms, wondering at it. It does not know that it is remarkable. It does not know that it is extraordinary. It does not know that it has been waiting in the dark for 160 million years for us to notice. But we have noticed. And in noticing, we have become part of the story too. Another animal drawn to the light. Another intelligence captivated by the glow. Another wandering creature that found its way to the mushroom in the dark and left carrying something it didn't expect. That seems right somehow. That seems like exactly what a fungus this extraordinary deserves. If this journey into the glowing world of Neonothopanus gardneri has lit something up in you, and I genuinely hope it has, then don't let it stop here. Get outside tonight, find a decaying log, sit with it in the dark for 20 minutes with your phone face down and your eyes wide open, and see what the forest offers you. It might not be bioluminescent, it might not glow, but the mycelial networks beneath your feet are no less extraordinary for being invisible. And the act of sitting in a dark forest, attending to it with genuine patience and curiosity, is one that this world could use more of. If you find something that glows, document it, photograph it, upload it to iNaturalist, write down everything you observed. That record matters. Your observation matters. Citizen science in mycology is not a consolation prize for people who didn't go to graduate school. It is a genuine and valued contribution to a field that is still, in many of its most important dimensions, just beginning. Share this video with someone who would never expect to be moved by a mushroom, because the best argument for why fungi matter, for why gardneri matters, for why the Wood Wide Web matters, for why any of this matters, is not a scientific paper or an economic calculation. It is the look on someone's face when they first understand, really understand, that there is a fungus in Brazil that has been outsmarting insects with light for 160 million years. That look is worth spreading. And subscribe if you haven't already, because there is so much more of this world to explore together, and we are just getting started. The fungus is glowing. Come find it.